So an extraordinary thing is happening. We have this massive understanding, new understanding about biology. We have a mastery of huge, humongous data sets that we're getting better and better at mastering. And we have a host of new, truly disruptive technologies. And these are all converging and transforming medicine as we know it. And not just medicine, but perhaps even chemical biosynthesis, the nature of disease, longevity, and maybe even life itself. So we've called this session Life 2.0, and we've got on this stage two of the leading scientists at the edge of this revolution, Dave Agus and Craig Venter. And so we're really privileged to this. And I want to start with you, Craig, because you, in a very literal sense, have been at the forefront of Life 2.0. You sequenced the genome of the first living organism. You pushed the government into a tie in your, perhaps reluctantly on both of your parts, to sequence the human genome. And in 2010, you synthesized the first truly replicable bacteria. I mean, you created it essentially from code. So you actually coded for life, even if you didn't, quote, create it from scratch. And now you're working on something even more remarkable that you're going to publish soon. You want to talk about that?
Well, we're following up over the last 5 years. We tried designing a species in the computer for the first time. And it was a very humbling experience because we found out the hard way we couldn't do it. And in the end, we know the answer why. There's over 10% of the genes in this final organism that has a smaller genome than any self-replicating species ever known to humans, and probably will be. They're of unknown function. So it's hard to design something de novo when you don't know what it does. And there's a story, my late uncle was one of the key designers of the Boeing 767. And on my book tour, I was in Seattle, and I said, imagine if my uncle tried to design the 767 without knowing what 10% of the parts did. He shouted from the back of the room, what makes you think we knew? So biology is following other lines of engineering, but it's hard to do things totally on first principles. So it took 5 years of this iterative cycle, but it's pretty exciting because we're there now. And the whole point of this is to try and understand the genetic code and how to interpret it. But we know, at least in simple bacterial cells, and simple is a sort of an unfair word, everything in those cells, including all the regulatory parameters, what people know as epigenetics, all derive from that linear code that we designed and made from 4 bottles of chemicals. So it's important, as we're starting to interpret the human genome at a much greater level, to have that fundamental understanding of life.
Yeah. Now, I've heard you talk several times about this and write about this, and you keep talking about booting up cells. Yeah. And I wonder, when it doesn't work, you can't call tech support, right? I mean, this isn't like, there isn't like an 800 number. How do you figure out what goes wrong when it doesn't boot up?
So, as I said, it's an iterative cycle. So if we can't get a living cell out of it, we tried adding back a few more genes in a random fashion, adding big cassettes until we could get life again, and then we went back to reducing those. So it was a cyclic set of experiments. That's why it took a team of 20 scientists 5 years to do this.
Wow. Yeah.
But it also puts into place this notion of designing pathogens or complex organisms. It was really damn hard to get this first really minimal cell. Designing something purposely would be very tough to do.
And it's great you use this word design, because it brings me to my friend Dave, who has written 2 best-selling books that are absolutely terrific, The End of Illness and A Short Guide to a Long Life, and a new one, which I've just read last night. I finished it, The Lucky Years. Thank you. And in it, you talk about that we have all of the information in front of us now to design our own health. Can you talk a little? I love that phrase, but talk.
The old days, you went to your doctor, and here's who drew a tube of blood, checked your blood pressure, weighed you, and then a couple days later called you with the data. Well, we now have the ability, and we heard Elizabeth Holmes, and we have the ability of gathering all that information before we go, whether it be Craig sequencing you, or whether it be all of your analytes, and actually sitting down and using that information for power. You are in charge now, and all of a sudden, the data's here. The challenge is how to use it. The challenge is doing something and having the context to what the data means, because data without context doesn't mean that much.
Yeah. Now, talk to me a little bit about, rather than, say, designing in the sense of picking and choosing what genes or mutations to sort of genetically engineer out or in, you talk a little bit about the ability of the body itself, that we have these anti-aging mechanisms within us. And you have a great story about a woman named Wanda Lunsford. Can you just talk about this, who this woman is?
Who?
Yeah.
It's the most amazing story in science I've ever seen. So in the 1950s, this graduate student in New York did an experiment, and it turned out to be the only paper she ever published in science. She took 2 mice, an old one and a young one, and she put them to sleep and tied their skin together. So after a couple days, the blood supplies joined, and then 3 weeks later, she looked, and there were new neurons growing in the brain, the heart was stronger, the muscles were larger, and she claimed she reversed aging. People actually thought she was crazy and called her the Dracula, the vampire, and all those crazy names. Well, just this last year, independently, a lab at Stanford, University of California, San Francisco, and Harvard repeated it, and it worked. And what they showed is there are proteins in the young blood that activate stem cells that get quiescent in you and I. So at age 25, they kind of go to sleep. And then once you turn them back on, the cure for many diseases is within us. So there's a clinical trial now to accelerate bone healing in people with fractures with these proteins. There's a trial now in neurocognitive decline. We're doing it in cancer. Right? Cancer in kids is curable almost 90% of the time. Once you turn 25, that same cancer is incurable. So if I can convince the body it's young, maybe I can have more of an impact on cancer.
Craig, we've even seen it with immuno-oncology, where we're untraining the immune system, or we're taking the brakes off of our own immune system in order to, harness its power to fight cancer and to recognize these malignant cells. What other natural abilities do our bodies have to heal themselves?
Well, the immune system, we're finding out, is probably one of the most important in cancer. And part of what we can do now at HLI is, aside from sequencing all the genes that change in you that can lead to cancer and the tumor that directly cause the cancer, we sequence the entire T cell repertoire at the same time, so we can see if your immune system mounted a response against these neoantigens in the tumor. And quite often they don't, which is why there's probably a tumor in the first place. If you've got a great immune system, you probably won't get cancer, or you'll be able to really diminish its effect. So understanding whether there's an immune response really helps with selecting some of these inhibitor studies where you can try and activate the immune system. But if there wasn't an immune response to begin with, the checkpoint inhibitor trials don't work as well. So we can actually now make new vaccines specifically for that individual and their cancer from these genetic changes that create new proteins in the tumor cell. That's how our immune system recognizes them in the first place.
Now, there was so much excitement when the human genome was decoded. And many people said this is the code of life. And what we're discovering is more and more there are more than one code of life. There are lots of little codes, and we talk about we're starting to discover what microRNAs might do, and we're looking at, from the DNA in the mitochondria, we're looking at epigenetics, we're looking at other things. In your mind, how many of these undiscovered codes are there? I mean, is this a...
Well, that's the cool thing, is right, we're learning a new dimension to each of us every 6 months. Yeah. So the role of the microbiome, you have tenfold more bacteria in you than human cells in you. And we're learning what they do. We didn't know about it several years ago.
Right.
All of a sudden, we're learning that we thought junk DNA was junk. It's actually doing something. We thought changes, polymorphisms didn't mean anything, and now we know they do. So these new dimensions make us complex.
Right.
And it means that we're probably never going to totally understand disease, but you don't have to understand a complex emergent system to control it. And that's one of the challenges. An amazing trial was done where they took 20-year-olds and they gave them artificial sweeteners because they were the greatest food ever, right? They hit your sweet tooth, yet they were not absorbed, so there are no calories. So they were perfect. But they gave them to them every day, and after a couple of weeks, they all had diabetes markers.
Wow.
Then they gave them antibiotics first, no diabetes.
Wow.
So what the artificial sweeteners did is they changed our microbiome to push our system toward diabetes. Even though they weren't absorbed, they had a dramatic impact on a disease.
Greg, we talk about, in all of these conversations we've had basically all day, we've talked about the problems of big data and too much data in many of these conversations in terms of business. Now, you live and die with the big data problem all the time. I mean, you have more data. I mean, it's almost immeasurable, the kind of data sets that you're working on.
So in fact, it's a challenge, but it's essential. So these same issues came from when we had the first genome 15 years ago. Yeah. There was nothing to compare it to. The same issue he was talking about a reference. So we all vary about 3% out of our 6.4 billion letters that we get from both our parents.
Right.
And so the only way to understand those is having very large data sets. So we're trying to generate millions of genomes, but in every case, so we have over 20,000 now, but we have phenotype and clinical data on every one to compare things to, so we can find the fundamental changes. So it is a big data problem. We're using Amazon Cloud, and we just punched a big hole in the cloud because we're in the 1% user group for Amazon, but most of the users are video streamers, not people who need lots of CPUs for calculations.
So big data has an ozone layer.
So it's
so
definitely an ozone layer.
Yeah.
So there's pornography and there's DNA.
And there's all right, that's good.
So we're now moving ahead of pornography and movie streaming. So, but it is a challenge.
So we've got a really cool group here, and I want to make sure that I throw it open to lots of questions here out in the audience. So please think of one while I ask this next one. So what a lot of people may not know about Dave is that he is a proteomics scientist. So as large as we think about the size of the genome, the sort of proteome is that much larger. I mean, a factor of what?
Well, I mean, it's order of magnitudes larger because
Right.
DNAs can encode for many proteins and the proteins can be changed. But, there's a conversation happening in our body all the time and we've never been able to look. And now through proteomics, you can actually listen to that conversation.
Right.
And you can start to know what's going on. So the way I look at it is, you're going to go to 2 Chinese restaurants and look at the ingredients list. They're exactly the same, but one food could taste good, one could be bad. Well, DNA is your ingredient list. What the food tastes like now is going to be the proteome. And so we now have the ability to interrogate that. And then they're going to be the metabolome, and then the microbiome. All these ohms together will start to give us a big picture.
Okay, let me see if we've got some questions out here. I'd love to get somebody's voice in here. Anybody? Question? We got somebody? Was that Miguel? Was that a question from you? No? All right.
Well, while you're looking for one, let me tell you how we're trying to attack data. We hired Franz Ock out of Google that many people know his work. He designed and developed Google Translate.
Yeah.
So we're using machine learning to try and understand and interpret all this data because it's a complexity that mere mortals sitting in front of a computer staring at data aren't going to find these associations. And so we're starting with a novel approach of we can now generate a photo of you just straight from your genetic code. And we think this is important as we interpret people's genomes. If you can see it can generate an actual photo of you and an accurate description of you. Also voice, so just from your voice print, we can tell your age, your sex, and your height.
Right.
And these features correlate with features in the face. So we're getting more and more correlations, but it's using this complexity of machine learning, not people trying to find, does this snip match your shape of your nose? And it's made an amazing progress in a short period of time. So we'll be publishing this shortly, but it has society implications because it means you can't just have your genome on the internet and be anonymous.
Yeah.
So whether it's DNA from a crime scene or because your DNA is on the internet, we can generate a photo of you and with photo imaging, find out who you are very quickly.
Right. We have a question right there from my colleague, Brian O'Keefe.
Brian O'Keefe from Fortune. With all this research you're doing about longevity, extending lifespans, it's going to have all kinds of effects on financial planning, how long people work, all this kind of thing. I'm wondering, like, how soon is an extreme lengthening of the average human lifespan, and what's the right length of a human average lifespan?
You want to answer the last question first?
Well, you know, when I talk about prevention in big settings, people come up and say, listen, pension plans plan for lung cancer, pension plans love heart attacks. You know, you have to, if we get everybody to live until their 9th or 10th decade, we're going to have a problem. But I truly believe is that with current technology, everybody has the potential to living till their 9th or 10th decade. And certainly if we can eliminate and prevent or delay most diseases, which we can, through very inexpensive means, we can have a big impact. Remember, there's a pill a day. If you take it, reduces not the incidence, but the death rate of cancer by 30%, heart disease by 22%, and stroke by 17%. And this pill is 2,433 years old. It's called a baby aspirin.
So human longevity is taking the approach, we're not trying to lengthen human life, but we're trying to lengthen healthy human life through these prevention measures. And so with preventative medicine, whether it's predictions from your genome, or if you come to our health nucleus where we measure whole body MRI imaging, 4D echocardiograms, DEXA scanning, we can give you the most complex picture of yourself to use as a reference for future changes. I'm not sure it's the right thing to try and get people to live to be 120. I usually get asked about this, what happens if you make people live to 200? I said, that's fine, but we should castrate all of them. So that might be a deterrent for some, I don't know. But we'll wipe out the planet if we change human lifespan to 200 years with our current standards.
When we think about codes, we think about linear relationships. And what we've discovered since the decoding of the human genome is that this is as much an emergent dynamic relationship between our genes and the other players and what we finally see as phenotypes. I mean, what really surprised you most in the science that you've done since the work you did decoding the genome?
What surprised me most in the last year is that we can actually accurately predict your age from your genetic code. If you'd asked me 10 years ago, I would have said that wouldn't be possible, and we'd do your genome once unless you have cancer. So our genetic code is constantly changing. Men in the age of the group up here start losing their Y chromosomes in their 40s and 50s. Women start to lose their X chromosomes. We start to get a lot of mutations in the mitochondrial genome. We can measure changes in telomeres, but we start accumulating mutations in cells. If you get too many, you get cancer. So this is now all measurable as we go forward. So I think we're going to understand aging at the molecular level, and hopefully if we take young blood, we'll find out how to reverse it.
Dave, what surprised you?
Well, you know, there's an amazing paper that came out recently where they took your eyelid. You know, your eyelid's apart, you normally put sunscreen on it, so there's a lot of UV damage. And when they looked, all of the changes that we classically associate with cancer were in the cells there, yet they didn't have cancer. And so it's the notion that DNA changes are necessary, but not sufficient. And so it's this, you know, the seed doesn't always grow unless the soil is right. And so the challenge is defining that system that would either push us toward disease or away from disease. To me, diseases are verbs and not nouns.
Right.
They're cancering. So I want to keep you in that health state. So even if those molecular changes happen, the cells don't change and grow.
Yeah, I love that phrasing. You talk about cancering and the sort of development of all these chronic ailments that we're used to. We think of some diseases as acute manifestations of something gone wrong. But for you, many of these things are dynamic processes. And Craig, do you sort of view this the same way about life and pathology?
Well, so cancer is a disease of the genome. It's accumulation of genetic changes. You cross over that threshold, all of a sudden you have tumor cells that grow uncontrollably. And the main way we can tell that by sequencing the genome in the tumor, comparing to the genome of the patient, we can find exactly what changed. And that's what's driving new therapies right now, and being able to understand what drug to use and what one not to use. And we're finding, particularly with HPV-16 caused head and neck cancer in men in their 50s and 60s, it's becoming almost pandemic right now. We see where the virus inserts in the genome and causes the cancer. So if you find early changes, or what we find in a lot of people is predisposition to cancer, and from the microbiome we find HPV-16 present. Right. The vaccines approved for young girls and young boys, it hasn't been approved for men in their 50s or 60s to try and prevent head and neck cancer. So these new findings may lead to new approaches.
All right, we've got a question right here in the front. Could you identify yourself too?
Yeah, Helmut Ludwig from Siemens. I always thought my good German sausage is really super healthy, and I now learned that there's processed meat in there. Today I learned that artificial sweetener is not that great either, which we probably knew before. So what do we do tonight when we have dinner and think about the long, healthy life that you promised us? What shall we focus on?
Drink lots of wine.
I like that idea.
It'll make it all seem better.
You know, there's a dirty word in genomics, heterogeneity. So when we think about the cancers that are really still so vexing to us, the ones that are, you know, responsible for most of our mortality are several of the solid tumor cancers, pancreas, lung, liver. You know, we see this enormous melange of genetic destruction where you could sample one little part, one cell over here, and you'll see some, you know, signature profile of genes and mutations, and it'll be completely different if you look somewhere else. And so how do you deal with that? And you deal with this every day, don't you?
Yeah, I mean, it's a major problem, obviously, is, you know, all these remarkable molecularly targeted therapies, they work, but they work for a very short amount of time. Mm-hmm. And so, you know, the challenge is making a major impact on a disease, and we're not doing that.
Right.
We've got these drugs approved now for $100,000 a year that give you an extra 2-3 weeks.
Right.
And, you know, they're done, I mean, this is what people don't get. They're done in randomized placebo-controlled trials. And in that setting, they make you live longer.
Right.
Problem is, in general, they slow the growth of the cancer. So if I take an x-ray of you today and your cancer is 4 centimeters, and I give you one of these fancy drugs, and then it's 6 centimeters in 3 months, did it work? Would your cancer have been 10 centimeters?
Right.
I have no idea. So I am flying blind almost all of the time when I'm treating these patients, and that's what doesn't totally make sense.
Right.
At the same time, heterogeneity is there. But if I look at a patient I treat, I almost never have one lesion respond and another not. They all synchronously respond and synchronously progress. So there's also crosstalk between all of them.
Huh.
So we're learning this new complexity. There are clues there. You know, the coolest paper in cancer came out this week. It is wild, where they looked at elephants. Elephants are 80 times bigger than most of us, and yet they have a much lower rate of cancer. And there's a particular gene, P53, that's one of the guardians of the genome that corrects DNA. They've got 20 copies, and we have 1.
Right.
All of a sudden, this outlier taught us one of the fundamental areas of biology, and it's staggering. With all that's happening now with technology and electronic health records and data, big data, we have to learn to look at the outliers and learn from them.
So some people would argue they're just on a vegetarian diet. So.
Well,
I'm kidding.
But you've looked at your own outliers. I mean, one of the remarkable things is that, of course, the genome that was decoded, the first human genome that we know about, was your genome, is your genome. What did you learn about yourself? I read somewhere that you have wet earwax.
Yeah, that was one of the early headlines in Canada.
Yeah,
It was,
right.
you know, unless you know there's different types of earwax, that doesn't seem so significant.
Yeah, so that probably wasn't as traumatic for you.
But I learned that I had a increased risk for heart disease. So 15 years ago, I started taking a statin, and I've outlived my father by 10 years, so I hope to take it further. But no oncogenes mutated, so low risk for cancer. I think one of the most interesting things, I was heterozygote for ApoE4 linked to Alzheimer's disease. Huh. And a lot of people are told this, and they think it's therefore a diagnosis. And the trouble with the early genetic tests right now, such as BRCA1 and BRCA2, your actual incidence is somewhere between 0 and 100% of getting breast or ovarian cancer. Those tests don't tell you anything except you have a slightly increased risk. Unless you know that you have a family history, then your risk goes up. So we're trying to sort out what makes sense across the whole genome. Mm-hmm. So I had early tests with MRI imaging and found no amyloid whatsoever in my brain. And then it turns out the ApoE4 allele may be associated more with preventing dementia following brain injury. So maybe an important screening tool for football players or others. So a lot of these things are being investigated right now as we can look at the genome, we look at the progression to disease, or more importantly, why do people with these alleles not get Alzheimer's? Why do 50% of women with BRCA1 and BRCA2 changes not get breast or ovarian cancer? It's because they have other genes that protect them. We've been looking under the lamppost and missing most of the picture.
You know, I want to just have one more question. Oh, no, we have a question right here. Great.
Hi, I'm Surya Gooding from Shopcake. Question about the lifespan. What do you see as the theoretical limit of how far you can take this, or us as researchers, how far can this go? What's the theoretical limit? And if we're not going to die from cancer, what are we going to die from? What's the limiting factor if that got eliminated?
You know, one of the great thinkers in our time was in the 1920s, Haldane. And they asked him, what's the limit on life expectancy? And he said 93. You know, if you asked us today, we would certainly say, you know, in the hundreds. But you start to get general engineering failure. You know, we've all seen it, right? People can be in 90s or 100. They all of a sudden, they can't do that much. They're not enjoying life like they did. They're starting to get general engineering failure without disease. And being able to reverse that, that is re-engineer the intricacies of all the blood vessels, re-engineer the brain, other things, is probably not going to be possible. But if we could just eliminate disease or delay disease till that 9th or 10th decade, we can all have a quality of life that we're proud of.
I think there's no hypothetical limit. And every time we set a limit in our minds, whether it's 93 or any number, if we learn how to repair DNA in early stages to prevent some of these changes, there may be surprising upper limits, but we need to make sure we can support extra long life on this planet.
You know, when you do your sequencing, you've of course living Moore's law, you know, the ability to sequence more and more and more. What we have with the price of so much of this healthcare provision is an anti-Moore's law. Why isn't the price of our drugs, you mentioned these outrageously priced drugs, why aren't they following these interventions?
Because it's a freaking monopoly. When you've got a monopoly, people price however they want. When you have a new drug for liver cancer, third-party payers have to pay because it's the only drug and it's FDA approved for that indication. You charge whatever you want. And when you have a monopoly, and we've seen cases clearly over the last several years, people taking advantage of that.
All right, well, I'm afraid we're out of time, but the big takeaway is there's no limit to life. There's just a limit to how much you can pay for it to increase this. I want to thank my fantastic guests, Craig Venter and Dave Agus. Thank you, guys.
Thank you.
Thank you.